Today we discuss salivary glands, and this is the last lecture of this semester. You know that salivary glands are exocrine glands in the mouth, and they produce saliva, which has digestive, lubricating, and protective functions. Very important. Saliva has its role in swallowing, in mastication, and in speech. Salivary glands. Let's speak about saliva function. The main function of salivary glands, it is the production of saliva, and saliva is over here are more. In detail, the composition of saliva. Saliva is over 99% of water, and a very small amount, yet the very small amount of additional inorganic and organic compounds, such as proteins. You can see enzymes are proteins and glycoproteins and other inorganic substances. Uh, these allow it to perform its main and important functions, as mentioned earlier. And these functions are swallowing, mastication, speech, and digestion, and lubricative, and at the same time protecting functions. A major role is related to the production of mucin, which acts as a lubricant during mastication. Mucin also aids pellicle formation and remineralization. The mucus film protects the mucosa and keeps it moist. You know that the function of saliva is also moistening of the oral cavity, oral mucosa, because oral mucosa is wet, moistured mucosa. Saliva brings substances into solution so that they can be tested. It also limits the activity of bacteria by causing the aggregation of these bacteria. And saliva contains minerals and acts as buffer. Both features help to maintain the integrity of the dental enamel. Also, it contains peptide growth factors such as epidermal growth factor, nerve growth factor, and they are produced by the submandibular gland, and their precise functions are not known for epidermal growth factor, because it may be involved in wound healing and maintaining gastroesophageal epithelial integrity. Saliva contains small quantities of numerous other proteins to which many different functions have been ascribed, such as among these are cystatin, inhibitor of harmful cysteine proteinase produced by bacteria and neutrophils, lysozyme, you can see lysozyme, defensin, histatin, lactoferrin, which are antibacterial agents, and also gustin, which is for detection of taste. And there are also, you can find here the amylase, maltase amylase, which cause the splitting of carbohydrate. And also it can be found the milk lipase, which splits the milk components in the infants. And also you can find here lysozyme, this defensin, lactoferrin, and other immunoglobulins, such as IgA, which are produced by plasma cells within the stroma. Stroma, you know that this is the connective tissue part of the gland. Also, polysaccharide hydrolyzing enzyme, this is amylase, alpha amylase, which is present in saliva, and begins the digestion of carbohydrate into the oral cavity. And also there is the kallikrein, which is serine protease and blood group substances. You can find the albumin, mucin, and albumin blood group antigens, and also you can find free amino acids and non-protein nitrogenous substances, urea, uric acid, creatinine, and also you can find here the inorganic substances as oxygen, carbon dioxide, nitrogen, bicarbonates, bromide, chloride, fluoride, phosphate, sodium, and calcium. And the varied functions of saliva can be taken for granted that it is only when salivary production and flow are disrupted that is true importance to the general well-being of the individual is realized. And with the aging in aged population, this is of potential clinical importance. And you know that inadequate saliva production leading to dry mouth of xerostomia can be caused by various factors affecting the major salivary glands, such as mumps, viral infection, radiation of the glands, or the normal side effect of drugs such as antihistamines. Let's now discuss salivary glands. Salivary glands may be classified according to size. You know that there are major salivary glands, three pairs of major salivary glands, such as parotid glands, such as submandibular glands, sublingual glands, and there are also minor salivary glands. And the type of these glands are exocrine glands, and you know what does it mean, exocrine glands. Here you can see exocrine glands. These are the glands which maintain the connection with outer epithelium. They are opened into the outer exterior. These are exocrine salivary glands. And also the types of secretion, there are mucus secretion, there is the serous secretion or mixed secretion. The three paired major salivary glands, you know, the parotid, submandibular, and sublingual glands are, and the numerous minor salivary glands are scattered throughout the oral mucosa and include the labial, buccal, palatoglossal, palatal, lingual glands. Salivary glands are not present in the gingiva or the dorsum of the anterior two-thirds of the tongue. In general, the parotid gland will synthesize more protein than glycoprotein and is a serous salivary gland. Consequently, parotid saliva has a lower carbohydrate content, whereas the submandibular and sublingual glands synthesize and secrete greater amounts of glycoproteins than protein. Then salivary glands are how they arise. You know that this is the salivary glands arise from a cord of epithelium growing into the underlying connective tissue, and the cord later forms the tube. At the end of this tube, a cluster of secretory cells forms such clusters form a structure which resemble a bunch of grapes and is called acinus, or this is single and plural is acini, and this is called end piece that are round or tube-like, and you can notice that here you can see here that these salivary glands are compound. Here you can see these are simple salivary glands which are tubular, and this tubule may be straight, may be coiled, may be branched, and alveolar. When there is a alveolus at the distal part, and these alveolus may be branched. These are simple salivary glands, but simple glandular glands. But salivary glands are compound. Compound means that they have as tubular as more than one tubule, and you can see that these tubules are unite into the one duct. And here you can see these tubules, and you can see also a scenery portion. Compound, yes, this refers, term compound refers to a fact that the salivary gland has more than one tubule entering the main duct. Tubuloacinar, you can see here, this is tubuloacinar, describes the morphology of the secreting cells. And there are some types that it is exocrine. Exocrine means, you know, that the surface of the tube, the duct of this gland opens into the outer environment, external environment, and it has maintained the connection with outer epithelium. And there are some types of secretion. There is the merocrine secretion. This is the secretion when secretion releases products, usually containing proteins, and by means of exocytosis at the apical end of the secretory cell. And most such exocrine glands are merocrine. But here you can see the other type of secretion. This is holocrine secretion, when this secretion is produced by the disintegrating, here you can see disintegration of the secretory cells themselves as they complete their terminal differentiation, and which involves becoming filled with the product. And the sebaceous glands of hair with your hair follicles are the best example of this holocrine secretion. And also you can see here, this is the apocrine secretion, when the secretion involves loss of membrane enclosed apical cytoplasm of the cells, and usually this secretion contains one or more lipid droplets. And apocrine secretion, along with the merocrine secretion, is seen in mammary glands. Now, let's discuss the structure, histological structure of these salivary glands. You know that these salivary glands, they have two main elements, and this is the glandular secretory tissue, which is parenchyma, and the supporting connective tissue. You can see, and you can see here, this is supportive connective tissue. It is encapsulated. It has the connective tissue capsule outside this structure, and this connective tissue forms the stroma. It is expanded into the parenchyma, and is called the septum or stroma of the capsule which surrounds the salivary gland. This stroma subdivides the gland into major lobes, and you can see this is lobe one, lobe second lobe, and here these are the lobes into the lobes. And the lobes are further subdivided into the lobules. Here inside the lobe there are some lobules into the, yes, into the lobules, and each lobe contains numerous secretory units These glands, they have secretory portion and conducting portion. Secretory portion are these tissues, secretory tissue, and conducting tissue. Conducting portion are the ducts. Each lobe contains numerous secretory units consisting of clusters of grape-like structures or acini, singular acinus. That means grape in Latin. And these cells are grouped around the lumen, and the secretory acinus may be serous, mucus, or mixed sera-mucus. Serous acini, and here you can see this is the serous, this is the mucus acinus, serous acinus, mucus acinus. The serous acini can be distinguished from mucus acini according to the nature of the secretion produced, and in structural terms, the morphology of their secretory granules. Serous cells secrete more protein and less carbohydrate than mucus cells. The acinus, why its lumen, you can see the acinus, why its lumen empties into the intercalated duct lined with cuboidal epithelium, and these cells, serous cells, are polarized. You can see these are polarized cells. They have a very small lumen. This acini. This is polarized cell. It is protein secreting cells, and usually pyramidal in shape. You can see it is pyramidal in shape with round nuclei. You can see now nuclei and well-stained rough endoplasmic reticulum and apical here. There are always, we see this micrograph, secretory granules, and these cells are joined apically by tight and adherent junctions. Serous cells form somewhat spherical unit, this spherical unit, and this is the acinus. Now let's see here. You can see this is the salivary gland, and it has this connective tissue capsule outside, and this capsule gives such expansions into the parenchyma, and this connective tissue part is called stroma. You can see the lobes, and these lobes contain a lot of lobules, and there may be intralobular ducts, and also here these connective tissue lobes, they carry nerve endings and blood vessels, and you see they are interlobal connective tissue which carries blood vessels, nerve endings, as well as ducts. Let's now describe the system of this duct system, and we can say that the most distally part of this duct system is intercalated duct. This is the smallest, nearest to the acinus. This is intercalated duct, and it leads from the serous acini here into the striated duct, and here you can see this is the striated duct, and this duct is usually compressed between the acini, and it is lined, here you can see, by cuboidal epithelial cells. The nuclei in the duct cell appear prominent owing to the relatively scanty cytoplasm. These are small cells. At the ultrastructural level, intercalated ducts are seen to consist of a simple cuboidal epithelial tube. Both luminal and the basal surfaces of the duct cells are smooth, and desmosomes unite adjacent cells. Here, these cells are connected, attached by desmosomes, and these cells sometimes contain apical secretory granules, and here you can see these apical secretory granules, and there are only small amounts of the organelles normally associated with protein synthesis. The intercalated ducts appear to contribute to the primary secretion, and there is moderate activity of respiratory enzymes in the intercalated ducts. You can see that as in the acini and also the submandibular glands, in the submandibular glands, several acini drain into the each several acini into the there are several acini that can drain into the intercalated each intercalated duct. And we can see here, you can see this is the intercalated duct, and two acini are drained into this intercalated duct, and this is the smallest part of this intercalated duct, and it contributes to the primary secretion of these serous acinas. Then the yes, the next portion of intralobular ducts is the striated ducts. And let's see here, these are the striated ducts. Why they are called striated? Because they are basal and hedge had such striations. These are the folds of membrane and is called striated duct. And for example, in parotid gland, intercalated ducts are characteristically long, narrow, and we'll see in my micrograph, and branching, and the striated ducts are intralobular. They are located in this lobes, intralobular, the striation, and intercalated ducts are intralobular, and form a much longer and more active component of the duct system than the intercalated ducts. The cells of the striated ducts have a large amount of cytoplasm and a large spherical centrally positioned nucleus that makes them easy to identify. The cells of striated ducts are highly polarized, and let's see here. Yes, these are the next. This is the striated duct, and it has larger cuboidal cells, and these cells are highly polarized. Their luminal surfaces have short microvilli from this side, and the duct's basal abluminal surface adjacent to the basal lamina separating it from the adjacent connective tissue shows numerous striations. These are the membrane foldings. Here you can see arrowed parts, such foldings, and these foldings are seen even under light microscope. And the ultrastructure here, you can see the striations correspond to multiple infoldings of the plasma membrane at the base of this cell. And there are also vertically aligned mitochondria, these mitochondria, and they are packed between the infoldings. Adjacent cells are intertwined in a complex pattern and anchored together by desmosomes. These cells are anchored by desmosomes, and this large surface area, supplied with high levels of energy from the mitochondria, is clearly involved in active transport of substances, and the striated ducts are the sites of electrolyte resorption, especially of sodium and chloride, and secretion potassium and bicarbonate without loss of water. This resorption is against a concentration gradient, and this is active transport, and it requires substantial amounts of energy, and this energy is provided by this great amount of mitochondria. Uh, the in the lumen, the material in the lumen is to convert an isotonic or slightly hypertonic fluid with concentrations similar to those in plasma into a hypotonic. It flows when these electrolytes, such potassium and chloride, enter by active transport into the lumen. This causes the osmotic gradient, and the water withdraws into the lumen. And there is therefore intense activity of respiratory enzymes in the striated ducts. Here you can see there are a lot of respiratory enzymes, and these enzymes, they maintain this concentration gradient and the hypotonicity of the fluid in the lumen, as also in the submandibular gland, in parotid and submandibular gland. The cells of the striated duct exhibit small secretory granules in the luminal region that may contain epidermal growth factor from this side, or other kallikrein or secretory IgA. The granules are less abundant in humans than in many other species. The luminal cells of the intercalated, striated, and collecting ducts contains low molecular weight cytokeratin intermediate filament number 7. These striated and intercalated ducts, they are intercalated ducts and striated ducts. They are interlobular ducts. And after this, the striated duct leads into the collecting duct, which is this is intralobular ducts, and they dumped into the interlobular duct. They are collecting ducts, interlobular ducts. They are collecting ducts, and whereas the intercalated and striated ducts modify the composition, make the saliva, make it more hypotonic, and maybe 10 the secretory ducts. They are not only ducts, this conducting portion of the system. They are secretory ducts. They modify the saliva. They are metabolic. They have metabolic epithelium. The main function of the collecting ducts is to transport it. To transport it. In addition to the columnar layer, you can see that these cells are more columnar layer, and this layer lacks striations. The collecting ducts situated in the interlobular region may have a layer of basal cells. basal cells. They are fewer in these basal cells. And this layer contains two layers. But when it enlarges, the main parotid duct appears like many excretory passages and contain two layers. And you can see that these layers are the other layer is the layer of basal cells. And what type of cells are these basal cells? These basal cells are a population a population of basal cells, which is in the striated and collecting ducts, and they are very sparsely distributed in the striated ducts and more densely distributed in the collecting ducts and especially in the lower duct, and in which they form a continuous layer. And here you can see that they form a continuous layer and the ducts pass further towards the hilum. These cells can be distinguished from other parenchymal cells by a combination of their morphology, their co-expression of cytokeratin 14 and the anti-apoptotic factor, and the proliferative index, which is about 3%, which is the highest of any cell in the region. And the basal cells have been implicated as potential stem cells and during turnover and the cell regeneration in salivary glands and during metaplasia when oncocytes and sebaceous cells can may appear. And however, some cell division is seen in the parenchyma of these cell types. Now let's describe the other type of cells, which is the myoepithelial cells. Here, let's see. Yes, these are the from outside. You can see this is the secretory cell. This is secretory portion which forms such acinas, and you can see the intercalated disc. This the duct. This is the most distally located duct, and it drains the secretion from these acinas. But here may be distinguished this type of cells, which are called myoepithelial cells. Myoepithelial cells lie between the basal lamina, and the basal membrane of the acinary secretory cells and the intercalated duct cells. Myoepithelial cells contract and they squeeze the acinas and aid in the secretion of saliva that has accumulated in the hollow center of the acinas. This cell helps move it out and into the duct system. These cells, you can see, they have such processes, and they are dendritic cells, called dendritic cells because they have such protrusions, and consist of a stellate-shaped body containing the nucleus and some tapering processes radiating from it. A different arrangement exists around the intercalated ducts, where the myoepithelial cells are elongated run longitudinally along the duct and have few short processes. Around the acini, these processes lie in gutters of the surface of the secretory cells, so the outline of the acinas remains smooth. Around the intercalated ducts, these cells lie more superficially and produce a bulge in the outline, and we'll see in an electron micrograph. Although salivary myoepithelial cells in some species stain positively for alkaline phosphatase, those in humans do not. They contain cytokeratin 19, and the myoepithelial cells contract as a result of activity of both parasympathetic and sympathetic autonomic nerves. And ultrastructure, the nucleus tends to be flattened, and intracellular organelles associated with the protein synthesis are not particularly abundant. The cell contains numerous contractile actin microfilaments, and the myoepithelial cells have desmosomal attachment. Here you can see, and here you can see attachment with underlying parenchymal cells. They have also gap junctions. They have gap junctions with the adjacent myoepithelial cells and hemidesmosomes attaching with the basal lamina. Um, the myoepithelial cells also contain cytokeratin intermediate filaments and contracting acting filaments, which can be used to help identify them from identify them from other adjacent cells by using immunocytochemistry. The presence of cytokeratin confirms the epithelial origin of the myoepithelial cells, and pinocytotic vesicles and dense attachment areas are associated with the part of the plasma membrane. And here you can see these myoepithelial cells and their numerous dendritic processes. They are staining positively, they are green, and these are the antibody to actin filaments, and they are around secretory acini, and these secretory acini are stained red. Then here you can see the acini. Here you can see the acini, and the you can see that this is the serous acini, and these are they have very small lumen. They are darkly stained, and this staining, if you compare with the mucus acini, they are darkly stained, and here you can see they have smaller lumen, they are serous, they are mucus, and these are the mucus serous or mixed structures. And such mixed structures, they have a mucus part, mucus portion, and such a crescent shape structure of serous part, which is called serous demilun. Serous demiluns, and they are mucus serous, and mucus cells. Such as goblet cells, they have, very developed rough endoplasmic reticulum, Golgi complexes, and they are filled apically with the secretory granules. And these granules, they contain heavily glycosylated proteins called mucin. When mucins are released from the cell, they become hydrated and form mucus, and most of the hydrophilic mucins are washed from cells during routine histological preparations, and causing the mucinogen granules to stain poorly and with eosin, and sufficient oligosaccharides usually remain, however, to allow mucus cells to be stained by the PAS, which is periodic acid shift method. Now let's continue our discussion, and here you can see the duct system, and this the smallest duct. You can see this is secretory portion and this conducting portion, and the smallest duct is intercalated duct, the most distally located duct, which drains secretion from this acini, and it leads to the striated duct. Here you can see these are serous cells. They are darkly stained cells, and here you can see the serous acinus, and each this is the serous demilune on the surface of mucus acinus, and you can see that these two mucus acini, they are drained into the one intercalated, and here three acini are drained into the one intercalated. These are very small intercalated discs, and they then drain into the striated ducts. And here you can see that there is then after this there is the interlobular duct, and here also there are the basal cells, which are stem cells which can proliferate, and they are involved in regeneration and repair of this duct system. And there, after this, there is the lobar duct with a collecting duct, which is only two layered or maybe stratified or pseudo stratified columnar cells. Here we can see, oh, this is the duct cell, striated duct cell. They have these special infoldings or striations, thereby they are called, the name implies. These striations are the infoldings of basement membrane, and here the very active process of active transport takes place, and a lot abundant mitochondria indicates a great energy needs of these ducts. And these ducts, striated ducts, they are secretory ducts. They are not simply conducting system, conducting tubes, but they are secretory. They have secretory function. They modify the primary secretion, and they modify the hypotonic saliva, which has its pH 6.5 to 6.9. Hypotonic. And here you can see interlobal excretory ducts, and it is sometimes, it has sometimes two-layer or two-layered. It is two-layered, and maybe a stratified columnar epithelium uh, before it reaches the outer surface. Uh, um, yes, the main duct carries the saliva to the mucosal surface and may be lined near its termination by a layer of stratified squamous epithelium. Here you can see these are the interlobal. Uh, um, this is the pseudostratified columnar epithelium, but near the surface, it may be lined near termination. It may be stratified squamous epithelial cells. Now about connective tissue. Septa, which carries blood and nerve supply into the parenchyma. Apart from fibroblasts and collagen, the connective tissue also contains abundant plasma cells, uh, fat cells. And you know that plasma cells, they secrete the immunoglobulins and are found in the stroma of the gland around the intralobular ducts. And with age, there is a decrease in a number of secretory cells, And unlike endocrine glands, the secretion of which is controlled by the activity of hormones, the secretion of saliva by the salivary glands is under the control of the autonomic nervous system. And what is the next? Yes, here you can see the serous acini stain darkly zymogen. They have zymogen granules, and the mucus stain slightly appears empty, cells filled with the mucoid materials and flattened nuclei. Yes, and the distinctive here, you can see that in mucus cells, the nuclei are basally located near the basement membrane, and they are flattened. In the serous cells, they are in the centrally located, and they are around round cells. And now on the micrograph, you can see the darkly stained cells are serous cells and serous acini. Here you can see these are the serous cells, and these are mucus cells. This is the micrograph of submandibular gland. And here, these glands, these spherical round glands with the centrally located nuclei are ducts, and they are striated ducts. And here and here and here and here, these are the striated ducts. And here you can see that this excretory duct is the here is illustrated. Now, yes, once more, we can see the serous cells, which are pyramidal cells. They have round nuclei. They are typical protein synthesizing cells. Why? They have well developed rough endoplasmic reticulum. They have a lot of Golgi complex, and they have a lot of granules in the apical part of the cell, and microvilli, apical microvilli. And here you can see this is the homogeneous electron translucent secretory granules, and the nucleus is flattened and has basal position. Here we can see this is the serous acini, these are the mucus acini, this is by hematoxylin eosin, and here is the bypass hematoxylin eosin periodic acid shift staining, which is special staining for mucus acini, and you can see their secretory mucus acini with strongly reddish color due to their high content of carbohydrates. This is the special staining for carbohydrates. Uh, what is it? Cerammucous acini, and yes, in glands that have both mucus and serous components, you can see such structure as mentioned, and this is it has the demilune shape, crescent shape, and it is called serous demilunes. And here you can see these mucus acini, they have larger lumens, they are larger lumens, these are mucus units, and you can see the serous acini, they have very small, sometimes the lumen isn't seen, and also on the surface of this acini, you can see the myoepithelial cells, and also here these structures are called serous demilunes. And here a lot of mucus acini, and on the surface, this is the serous acini. They are darkly stained. They have very small lumen, which is not seen very often. And you can see the here and here and here larger lumen. And on the surface, you can notice such structures which are called serous demilunes, and and it was thought that these are the artifacts in the histological slides and are not seen in the electromicrographs, but they, during preparation, they acquire such position, and they are still called cirrus demilons. And here is the structural and functional unit of salivary gland. Let's summarize, and you can see that there is the basal lamina, and between the basal lamina and the cell membrane, you can see the myoepithelial cell, and here myoepithelial cell on the collective intercalated duct cells, and also you can see the acinar cells, they structure this bunch of cells or cluster of cells, like bunch of grape is called the acinus. And you see that this intercalated duct is the smallest duct. It is the first tube of, which drains the secretion from secretory structure, secretory portion. And then you can see the striated duct, which has on its basal side, it has membrane infoldics that give this duct such striation appearance. And once more, here you can see the intercalated ducts are simple. They are lined with simple cuboidal epithelium, and intercalated ducts are the smallest ducts. Then are striated ducts. These are intralobular ducts, striated ducts, and they have cuboidal or low columnar cells with rounded nuclei. And there are these intercalated and striated ducts are intralobular ducts, and there is after this interlobular ducts, which are excretory ducts or collecting ducts, and they have columnar cells, such columnar cells. And there is also interlobal ducts, and they have pseudostratified columnar cells. And the main duct, which then opens on the surface, it has such stratified columnar, which then transitions into the stratified squamous epithelium. And here we can see this is the intercalated ducts. Intercalated ducts are shown in a longitudinal, this is the upper panel, the longitudinal section. And here you can see the cross section of this intercalated duct, and the diameter of the intercalated duct is considerably smaller than that of the end piece of the structure. The duct cells are low cuboidal nuclei of myoepithelial cells. Here you can see the nucleus of myoepithelial cells are present along the basal surface of the duct. Then let's now talk about parotid gland, and this is the largest gland. This is the largest gland, parotid gland, and is the um, here, yes, largest gland, and it is serous gland, and it is enclosed within the well-defined capsule. Maybe we have this picture. No, defined capsule. The acini of the gland are serous. Here you can see, although mucous cells are occasionally present, and this is the duct, intercalated duct. You know that in parotid duct, the intercalated ducts are longer. These are intercalated ducts in the parotid salivary gland. And these cells have a characteristic granular appearance with the routine hematoxylin and eosin. And here you can see this is the electron micrograph. And the connective tissue septa can be seen subdividing the secretory parenchyma into lobes and then further into lobules. Maybe here, yes, this is the parotid gland, and you can see this is parenchyma, and between these lobes, this is lobe, and it has some lobules, and you can see intralobular glands, and also you can see the interlobular glands, intralobal glands, and connective tissue which divides these lobes, and each lobe can have a lot of lobules. Yes, this is connective tissue. This is the interlobular collecting duct and interlobular artery and interlobular connective artery here, and this is the interlobular collecting duct. And also you can see a lymph node, and you know that in the oral cavity there is the diffuse lymphoid tissue, which is very strong defense mechanism in oral mucosa. You notice that the connective tissue contains blood vessels, nerves, and collecting ducts, and the lumina of the acini are very small. The Let's see here. Yes, and the prominent nuclei are round. Here you can see and located in the basal third of the cell, which is basophilic. The granular appearance, you can see the granules on the apical of serosacinar cells, results from the numerous granules in the luminal part of the cell adjacent to the lumen, and intercalated ducts pass from the acini and open into striated ducts. These are intercalated ducts, and here you can see connective tissue, septa, and also serous secretory units. These are the serous secretory units, intercalated ducts, and also you can see the blood vessels, which are numerous here in the parenchyma, in the connective tissue of the parotid gland. Here, this is once again, and these cells, they have the wedge shaped outline and the basal surface. Basal surface, you can see the basal surface. These cells, it shows the membrane, shows the microvilli and infoldics, and this basal part of each cell cells is delineated from the surrounding connective tissue by basal lamina, and also it contains the rough endoplasmic reticulum, and capillaries are seen in close proximity. The luminal part of the cell contains dense round secretory granules, and there are also canaliculi between the cells and join the lumen. And we'll see it in here. You can see this is a parotid gland, intercalated ducts, intercalated here. There are the intercalated ducts leading from serous acini and into the striated duct. And you can see here and here, you can see the fat cells. Here intercalated duct, here intercalated duct, here intercalated duct, and there are the cuboidal cells which line the intercalated duct here and here and here, and they have prominent nuclei here due to their scanty cytoplasm, and the nuclei in striated ducts are more centrally positioned within these cells. There, the parotid gland, it's has the last duct which opens on the surface is called the Stenson duct, and the Stenson's duct opens at the level of maxillary molars, and here you can see this Stenson's duct. And here is the parotid gland micrograph, and this parotid gland which contains serous and pieces, and these round structures are striated ducts, and you can very easily distinguish striated ducts by their round shape and centrally located nuclei. And here also, this is parenchyma of the gland, and here you can see the connective tissue septa in the gland. And here it is the dark granular appearance of cytoplasm of serous acini of parotid gland, and here is indicated intercalated duct, which is very often compressed by this acini. And here maybe this is the compressed intercalated duct, and here intercalated duct, and they are dumped into the striated duct. And here you can see in connective tissue septa the plasma cells. Plasma cells are abundant in connective tissue, and you know that plasma cells are very important cells which produce antibodies. And here you can see the serous cell, and serous cells abundant granules. You can see the abundant granules at the apical part of the cell, and you can see the lumen. Also, you can see the adjacent blood vessel with erythrocyte. It's seen at the basement membrane, and the canaliculi between the cells, and you can see very well developed endoplasmic reticulum and Golgi complexes. And this is the typical appearance of the cells which are actively involved in protein synthesis. And these adjacent cell membranes contact at the desmosomes. Here maybe desmosomes. And here you can see this is the brown staining, which shows the activity of the respiratory enzymes. This is the cytochrome oxidase in submandibular gland with moderative activity in serous acini. And here you can see that there is the intercalated duct, and so very intense activity in striated duct. And here this is the striated duct and the small intercalated duct. And this is the theme of parotid serous cells, which shows this is higher magnification, shows the secretory pathway, and you can see the mitochondria and a very well developed Golgi apparatus and the rough endoplasmic reticulum, rough endoplasmic reticulum, and also you can see the secretory granules, very dense, electron dense granules. And also here is the mature secretory granules, and here you can see the secretory granule discharging by exocytosis into the lumen. The next is transmission electron micrograph of serous of red parotid gland, and then the nucleus. You can see nucleus and see very well developed rough endoplasmic reticulum with Golgi complexes, and secretory granules, and lysosome, and also you can see a lot of canaliculi between these cells. This is the intercellular space, and a lot of canaliculi, which for the communication between adjacent cells. And now here also there is the well-developed rough endoplasmic reticulum with a lot of ribosomes, and here, and you can see these Golgi complexes and secretory granules, mitochondria, and here you can see immature granule which continue maturation. Now, the next type of here, you know that there is the next pair of salivary glands are submandibular glands, and the second largest glands are submandibular glands, and they produce a mixed mucoserous secretion, and they are called the mixed gland, and as parotid gland is the only serous gland. The submandibular and sublingual glands are ceramicus gland, and the overall mean of the proportional volume of mucous cells is about 8% of the total acinar volume, although it varies between 1 up to 33%, and the gland has a well-formed connective tissue capsule, and the serous cells, duct system, myoepithelial cells, and the basal cells are found as described in the parotid gland, and the intercalated ducts are shorter, and the striated ducts are longer and more conspicuous. And let's see, what is the predominantly serous acini, some mucous acini, and the serous demilus, and the short intercalated ducts, striated ducts with simple cuboidal lining epithelium. There are interlobular ducts with striated cuboidal or stratified columnar epithelia surrounded by connective tissue. And let's see, it drains saliva from the submandibular. This is Wharton's duct, which opens into the floor of the mouth at the junction where the front of the tongue meets the mouth floor. A capsule of tissue also surrounds each of these glands, which give off mixed secretion, mostly serous in nature. And here you can see this Wharton's duct. Here you can see this the submandibular gland, and here it has these lobes which contain a lot of lobules, and you can see this lobular organization of the gland, and individual lobules. Individual lobules are separated by connective tissue septa, and you can see the excretory interlobular ducts, excretory ducts, interlobular ducts. Interlobular means between lobules. And you also can see the blood vessels. Here you can see blood vessels, blood vessels, and nerve endings in between this. And here it is submandibular gland, and we now can distinguish the structures. Here you can see this is the serous secretory unit, and the second is the mixed secretory unit. You can see this is the mucous part of mixed secretory unit, and here you can see that it is this serous the milion, and here also you can see this is the mucous part of mucous part. These are mixed secretory units. And there is the here you can see intercalated excretory duct, and also you can find here this is the striated excretory duct. This is striated duct, and here you can see interlobular excretory duct, and this is longitudinal section, and this is cross section interlobular excretory duct. And maybe mucus part of the mixed secretory, and here you can see serous part of serous demilon. And here, this is the submandibular gland, and you can distinguish here the serous acini, and also you can see intercalated this round strap. And here you can see mucus acini, and here you can see this mixed ceramicus gland with its specific serous demilon. And here also you can see this serous demilon. And the serous demilon produce lysozyme, which is a bactericidal enzyme, and the secretion also contains water and salts. And here you can see this is light micrograph of serous end piece of the human submandibular gland, and which is the stained with toluidine blue, the apical cytoplasm of the serous here. Cells contain secretory granules, and here you can see these secretory granules of variable density, and also you can see the lumen. And also from outside, you can see this is the nucleus of a myoepithelial cell. And here you can see the intercellular canaliculi here between these cells, intercellular canaliculi in a serous end piece, and the lumen. Here you can see lumen and intercellular canaliculi. They are filled with India ink, and these arrowheads indicate intercellular canaliculi, which are extending between adjacent cells. Now, what is this? It is the mucus cell. And here you can see these are secretory granules, which are round, discrete. They have such light appearance, white appearance. And here you can see the serous cells, and you can notice the abundance of rough endoplasmic reticulum with lots of ribosomes. And this is aligned, these cells, they are aligned to surround the common lumen. This is the common lumen, and no serous demilons could be detected here. And what is this? Yes, this is the part of esinus of submandibular gland, which shows a myoepithelial cell. Here you can see myoepithelial cell with a process or dendritic process in which bundles of contractile filaments are seen here, and here you can see these filaments, and here you can see the serous cell. The myoepithelial cell embraces the underlying serous cell, in which variation in the appearance and density of secretory granules is seen. And here is the special staining, myoepithelial cell staining, positive brown for the antibody to cytokeratin 14. And you can see that on the periphery of this acini, there are these brown staining myoepithelial cells. And here, this is the mucous end piece. Here you can see the mucous end piece, and you can notice these structures, which are demilunes, and also you can see the lumens here. This is the flattened nuclei on the basal part of these cells. And here also you can see the serous acini here and here and here. Now, what is this? Yes, this is the terminal portion of mixed gland prepared by conventional immersion fixation in glutaraldehyde, and secretory granules you can see of mucous cells here are enlarged, and some have coalesced. Here they are merging to each other. And also you can see these serous cells, and they are compressed by the distended mucous cells toward the periphery portion of the acinas, and they form such serous demilune structure. And here, these are the submandibular gland, which shows darker serous cells. These are the serous cells, serous acini, and the light mucous acini. You know, submandibular gland is the mixed gland. It has serous acini and the mucous acini. And also you can see these crescent-shaped structures, and here and here, and here, and here. These crescent-shaped structures are serous demilunes, and here you can see the intercalated duct, and also you here, this is the striated duct, and you can find here in the connective tissue, there are the plasma cells. In connective tissue there are plasma cells, and also are find fat cells. What is this? This is the mucous cell, which is packed with secretory granules that contain stained glycoprotein and small unstained spherules that contain protein. And the adjacent serous cell shows scattered secretory granules. Here you can see secretory granules, composed of stained glycoprotein and unstained spherules of protein. And there is the next lingual next type of salivary glands are sublingual glands, and these sublingual glands are these sublingual glands, which are also they are the mixed glands, and they secrete saliva into the floor of the mouth. And this human sublingual gland is not a single unit. like the parotid gland or submandibular glands, but it is made up of a posterior part of the posterior part is the greater part, sublingual glands unit, that is not always present, and an anterior part which is the lesser sublingual gland and consists of the 7 to 15 small salivary glands, and each these salivary gland structures have its own duct system emptying into the sublingual fold, and it is Bartoli duct in a similar location to Barton's duct, and it is also mucocerous or seromucous gland, and the mucus acini are predominate, and some mucus serous acini. You can see that the mucus acini, and also there are seromucous acini with serous demiluns, and the sublingual gland was conventionally considered to be a mixed gland with predominantly mucus elements, and but in by conventional staining methods for light microscope, it is seen that it consists of many groups of pale staining mucus cells with darker staining here, so-called serous demiluns, and yes, the duct system is much less well developed than in the other major salivary glands, and striated ducts are usually absent. The acini sometimes lead to intercalated ducts, but these may be absent, and the acini lead to collecting ducts. Let's see, maybe we have yes, collecting ducts, which lack the basal striations that characterize striated ducts. The sublingual saliva is therefore rich in sodium. The greater sublingual gland drains into a main duct, and the individual glands of the lesser sublingual gland drain independently through many smaller main ducts that open directly into the overlying oral mucosa. And here you can see the pale staining mucus acini. Here you can see, and here, and also you can distinguish these plasma cells. Here there are plasma cells, and maybe here are plasma cells, and also you can find this is the serous demilon, and acini here is caused by the scarcity of secretory granules in mucous cells at the beginning of the secretory cycle, although parts of these cells show pale areas of secretory granules. And here it is the minor salivary mucous salivary glands. The minor salivary glands are classified by their anatomical location in the submucosa. They are buccal, labial, palatal, palatoglossal, and lingual. It has been estimated that they may number between 450 to 1,000. They are mucous glands, except for the serous gland of von Ebner, you remember from oral mucosa taste buds, which drain into the trench of the circumvallate papillae, and the labial and brachial glands here are illustrating here, and the palatoglossal glands are located in the region of pharyngeal isthmus, and you can see that it is a mucous salivary gland located in submucosa below the epithelium of the oral cavity, and saliva secreted by minor salivary gland reaches the oral cavity through short ducts. You can see this minor salivary glands, and they are located in a lamina propria of the oral mucosa, and you can see the stratified squamous surface epithelium. And what is there here? It is the intralobal striated duct. You can see this nice picture. You can notice that they are round, this on cross section round. They have centrally located round nuclei, and they also have this striation. They also have basal acidophilic striation. And what is this? This is striated duct, this is cross section, this is longitudinal section, and you can see the small blood vessels here. Notice small blood vessels are present adjacent to striated ducts. Maybe here it is the collecting duct. Collecting duct, and you know that this duct may be two layers of cells. And here, what is it? The intralobular and interlobular ducts. And here you can see that these are the mostly serous lobules, and here you can notice that this is the intralobular duct, and again you can see the mucous lobules, and also can be found serum mucus acini. And here, what is it? This is the excretory duct, this largest duct. Excretory ducts are located in the interlobular connective tissue septa, and the smaller excretory ducts are lined by a pseudostratified epithelium and have a larger lumen than the intralobular striated ducts, and numerous small blood vessels here surrounding this duct. Small blood vessel, blood vessel, blood vessel. Numerous small blood vessels are located adjacent to the duct. And you can also see the nuclei of dendritic cells. Here you can see, and here you can see dendritic cells are indicated by the arrows, and the larger excretory ducts may have a stratified cuboidal or stratified columnar epithelia, or and when they are near to the surface, they become stratified squamous epithelium. And here you can see that there very often there are the structures, newly formed structures. You know that there are a lot of electrolytes in the saliva, and they can form the salivary calculi, or stones may obstruct salivary flow. And here you can see schematically the stone which is in the duct and can cause such, you can see this is Wharton's duct silolith. The structures are called siloliths, and here you can see this silolith, and here you can see the removal of this silolith, and this is about 1 centimeter, and it usually occurs in the submandibular gland because of the tortuous course of the duct and because of the viscosity of the secretions. And and acute symptoms occur during a meal time. And here you can see the silolith, which obstructs the salivary stone, and it is the most common disease of the salivary glands in middle-aged patients, and more than 80% of the salivary siloliths occur in the submandibular, and you can notice how large this stone is. And 6% to 15% occur in the parotid gland, and about 2% in the sublingual gland. And here you can see the silomicroliths. These are small, hard masses, concrements that are only evident microscopically in the salivary glands and may occur in the parenchymal cells, in the lumina or in the stroma. And here you can see these cylomicrolids occluding the lumen of striated ducts, and this has led to the formation of inflamed atrophic focus and here here you can see fat cells and secretory inactivity leads to an increase in cylomicrolids and allows microbes to ascend the ducts and settle in the atrophic foci out of which microbicidal saliva. Then here you can see this autophagosome in an acinar cell in which crystals of hydroxyapatites here and here and here are being deposited in secretory material associated with membranous debris form cylomicrolids. And here you can see the structures which are called mucocele. And this mucocele is the damage of the duct of minor and sublingual salivary glands and may result in the extravasation of mucus into the surrounding soft tissues. And when the extravasation persists and extravasation mucocele is formed, and here you can see, and in the case of the sublingual gland, it is also termed the ranula, and you can see why it is called the ranula and ranula on the floor of the mouth because it resembles the belly of a frog, and when a ranula is situated about the mylohyoid muscle, it produces a painless swelling here that may displace the tongue. And this is the another type of ranula and is dividing or plunging ranula, which occurs when a simple ranula ruptures, and the rupture triggers the formation of a pseudocyst that extends into the neck through a defect of the mylohyoid muscle, which is a group of neck muscles that control the tongue. This was the last topic, but I'd like to discuss the immune system of the oral cavity, because you know that the oral cavity and the nasal cavity, they are very important. They are large lumen openings in the body to the outside world. And the oral cavity is the entrance to the gastrointestinal tract. And the nasal cavity, you know, that provides entrance to the respiratory tract. And the gastrointestinal and respiratory tracts meet it, then cross over in the pharynx, and the oral cavity is greatly colonized by bacteria, fungi, and viruses, and the intake of air or food constantly brings new microorganisms into the oral cavity. And to control this colonization and defend against invading pathogens, the body uses a defensive system with external areas located in the oral and pharyngeal mucosa. You know that mucosa is a mucous lining, oral mucosa or nasal mucosa. They are the defense mechanism. They are physical barriers against invader. And the defense mechanism includes an innate immune system also known as the non-adaptive or non-specific immune system. And there are the special cells, immune cells, primarily macrophages, natural killer cells, neutrophils, eosinophils, and the basophilic granulocytes. Also, there are mast cells, which are connective tissue mast cells, and they are able to detect invading pathogens and render them harmless. Then changes to the body's cells can also be detected, although this non-adaptive immune system cannot adjust to new pathogens and the changes, it is nevertheless quick to react. It has this innate immune system. First of all, primary defense mechanism is this physical barriers such as skin, such as mucosa or mucosal lining, such as inflammation, for example. And this response is very rapid. Along with non-adaptive immunity, the body also has adaptive or specific immunity. And these cells which are involved in immunity can adapt to pathogens and cell changes and transmit the information about detected changes to very high number to other cells involved in this immune response. And you know that the key players of specific or acquired or adaptive immunity are T and B lymphocytes. These are the cells which are derived in bone marrow, but T lymphocytes in early stages of fetal development migrate to the thymus, while B lymphocytes are responsible for the production of fluid bound immunoglobulins. This is humoral immunity. T lymphocytes recognize cell surface proteins and hence are responsible for direct and indirect defense via cells, and this process is called cell mediated immunity. And after infection, specific antibodies and the memory cells are retained in the body, which speeds up the immune response when there is renewed contact with the same pathogen. And this is by the why the innate immunity has no memory, but specific acquired immunity has memory. And these T as well B lymphocytes, they form memory cells, and these memory lymphocytes live in our body for years and can be transferred by from mother to infant. All parts of the immune system are found free in the tissues of the oral cavity and are concentrated in the tonsils, salivary glands, lymph vessels, and the lymph nodes. You know that we discussed saliva and salivary glands, and saliva is very potent defense mechanism against invaders. You know that it contains such enzymes, bactericidal enzymes such as lysozyme, defensin, lactoferrin. Lactoferrin binds iron, and iron is very important element in bacteria's diet. And the decrease of iron's amount causes bacteria's death. And also, you maybe know that there is the lymphoid tissue which provides lymphocyte proliferation. You know that T and the B lymphocytes are only formed elements from blood, which can proliferate outside the bone marrow. And this lymphoid tissue, such as thymus, such as lymph nodes, such as spleen, they provide the site of proliferation and differentiation and conversion, for example, of B cells, B lymphocytes into plasma cells. But beside these structures, there is also diffuse lymphoid tissue, and this diffuse lymphoid tissue is the largest lymphoid tissue in the body, and it is located beneath the epithelium, beneath the mucosal lining of gastrointestinal tract, respiratory tract, in submucosa. These lymphoid tissue, they are not encapsulated, such as lymph nodes or spleen or thymus. This is diffuse without encapsulating, and they contain a lot of defense adaptive immune cells. But in the oral cavity, you know that there are tonsils, and the tonsils also are lymphoid tissue. What does it mean, lymphoid tissue? The tonsils are formed by the lymphoid tissue, and the main structural component of lymphoid tissue are reticular cells. There are such fibroblastic cells, reticular cells. They form a meshwork of reticular fibers, which consist of collagen fibers, and these three-dimensional scaffold houses the immune cells. There are the lymphocytes, B cells, T cells. Also, there are antigen presenting cells such as macrophages, such as dendritic cells, and maybe if there is infection, granulocytes like neutrophils, eosinophils, and basophils. And the oral mucosa is pervaded by a mucosa associated immune system that consists of immune cells migrating freely in the tissue, and epithelial and lymphoid cells that combine in the tonsils into a structural and functional unit. And they lie under the epithelium of the oral mucosa, where it is thin, and are known as lymphoepithelial organs and tissues. Isolated crypts containing lymphatic tissue, such as in lingual tonsils, in palatine tonsils, are occasionally referred to as cryptolymphatic units, although the term has not become established in areas of anatomy. And where the nasal and oral cavities lead into the pharynx, diffuse lymphatic tissue is found in the mucus of the pharynx, especially in the lateral walls, and the tissue concentrations in the unpaired pharyngeal tonsils, the equally unpaired lingual tonsils, and the paired palatine tonsils. And combined, this tissue and organ formations are known as the lymphatic pharyngeal ring of Valdier, tonsillar ring. The epithelium of the tonsils is derived from the embryonic endoderm, and before birth, lymphoid stem cells from the yolk sac, from the fetal liver, and soon as it is formed, from the bone marrow, migrate into the mesenchymal stroma. And these tonsils, they hold a large number of immune system cells, and thus have the functions of antigen recognition and the formation of immunocompetent lymphocytes, starting from an antigen that comes into contact with the body in the epithelium of the tonsils. These cells and the tissue structures involved in adaptive immunity and both these structures they form the specific immune, they frame the specific immune reaction or response to the invaders. Beside these specific cells, there is also the epithelium covering the tonsils is comparatively thin, and there are located in these epithelia. There are the other sides type of lymphocytes which are called gamma delta lymphocytes, and which forms the only 2% of lymphocytes, and they are called intraepithelial lymphocytes, and they predominantly are in the epithelial lining of mucous membrane of gastrointestinal tract, and beneath these cells there are also so-called M cells or microfold cells, which are the special cells of the follicle associated epithelium covering the areas of the tonsils, which contain lymph nodules, and they take up antigens from the oral cavity by endocytosis, and these cells are specific by their pockets on basolateral sides where can be these immune cells located, and they take these cells up antigens from the oral cavity by endocytosis, and these can be macromolecules, but whole bacteria, viruses, and smaller parasites can also be phagocytosed. And on their basal sides, the M cells bear numerous pocket-like imaginations, and as was mentioned, in which B lymphocytes, T lymphocytes, and interdigitating dendritic cells and macrophages are located. And in this way, they come into contact with the antigen, absorb it themselves, and receive information about it. After this, you know that macrophages communicate with the T lymphocytes by their major histocompatibility complexes. This is the complex, protein complex, on which macrophage displays the remnants of the pathogen, and the T cells only recognize this structure, this MHC2 complex with the antigen. And after this, T cell is activated and releases cytokines, and these cytokines activate the B lymphocytes, and the B lymphocytes begin to differentiate. Here takes place clonal selection, and only clone which produce these antibodies with high affinity to this pathogen, only this clone survives, and thereby it is called clonal selection. And B lymphocytes, they, after clonal selection, they give effector cells such as plasma cells, and they also form memory cells or memory B lymphocytes, which stay in body in blood circulation. And this is the potent immune mechanism in oral cavity and in oral mucosa. And as you know, all these cells, as leukocytes and lymphocytes, B and T lymphocytes, and also antigen presenting cells, are involved in this process. Thank you for listening, and this was the last lecture of this semester.
Thank you, doctor.
Yes, yes.
Thank you. Thank you.
Mhm. If you have some questions, you can communicate me. Yes, about the final exam. Maybe you are interested in this final exam. There will be the 40 questions, and predominantly the half of these questions will be from this embryological part. This is the early development of tooth, development amelogenesis, dentinogenesis, development of periodontium, development of cementum, periodontal ligament, dental pulp, and alveolar bone, salivary glands. For this part, the last five lectures, this will be the 20. Half of these questions and half will be from previous material. Any questions?
No,
If you
thank you.
Yes, if you have questions, you can email me.
Yes, so if I have any questions about the curriculum, I'll email you like tomorrow.
Please repeat. I don't understand.
If I have any questions, I can email
Yes,
you,
yes,
right?
yes. Of course,
Okay,
sure, sure.
okay.
Goodbye.
Goodbye.
Thank you. Bye bye.
Good luck. Good luck.